Hello. Sir, mic test chestara.
Yes, sir, it is audible, sir.
Okay. Is it audible?
Yes, sir. Yes, sir.
Okay.
It's clear, sir.
Yeah.
Distinguished professors and learned participants, a very good afternoon to all of you. I extend a warm welcome to the 5th day of 1-week online faculty development program on emerging trends in pharmaceutical sciences, challenges from academic research to commercialization. Let us proceed with the session 6 of faculty development program. I feel privileged to introduce the august speaker of today's session, Dr. Falguni Pati. I heartily welcome the meticulous, very knowledgeable, and the eminent speaker of today's session, Dr. Falguni Pati sir, Associate Professor, Department of Biomedical Engineering, Indian Institute of Technology, Hyderabad, to deliver a scientific talk on the topic entitled 3D Printing Technology in Pharmaceutical and Biomedical Applications. Let us listen to the brief profile of Dr. Falguni Pati sir. Now I request respected, admirable, highly knowledgeable, and very down-to-earth, incredible human being to whom we always look up, Dr. T.P. Rao sir, Associate Professor, Department of Pharmaceutics and Biotechnology, to present the brief profile of Dr. Falguni Pati sir. Over to Dr. T.P. Rao sir.
Thank you, Dr. Arifa. So it is my pleasure to introduce a true researcher like Dr. Falguni Pati from IIT Hyderabad. And I will give you a brief introduction about the Dr. Falguni. Dr. Falguni is an associate professor in the biomedical engineering department at IIT Hyderabad. He completed his PhD from IIT Kharagpur and had postdoctoral appointments in Prosthetic, South Korea and KTH, Sweden, prior to joining IIT Hyderabad in 2015. His lab at IIT Hyderabad, Biofabrication and Tissue Engineering Lab, focuses on developing innovative tissue engineered products and in vitro tissue organ models with 3D bioprinting technology. His research interests include developing novel bioinks, bioprinting methods, and protocol for the production of tissue analogs on demand and in vitro tissue organ models. Dr. Falguni designed and developed a course on biofabrication for the postgraduate students at IIT Hyderabad, which is the first of its kind in India. Great, sir. He is the recipient of many awards, including the prestigious Ramalingaswamy Fellowship in 2016. Dr. Falguni has filed a US patent and 5 Indian patents. He has authored over 30 journal papers and 10 book chapters, one book, and presented over 50 keynotes and innovated lectures. And also he's having many funded approved projects in this account. Great, sir. So really we are lucky to have you, a person like you. So please continue with your seminar. Thank you.
Thank you very much, Professor Rao. Thank you for the introduction and for inviting me to this forum for presenting. I'm audible, right?
Right, sir. Audible,
yeah.
Sir.
Yes, thank you. Yeah, that's all. Thank you very much for inviting me to this forum for presenting our research work. So today, mostly I will discuss, or I will introduce the topic that we do in our research in our lab, that is 3D printing. So 3D printing for pharmaceutical and biomedical applications, that's the title. Let me share my screen also. I want a confirmation. Can you share my screen? Is it full screen?
Yes, yes. Yeah, it's visible, sir.
Thank you. Thank you very much. Okay. Good afternoon, all the participants. I want to welcome you, all of you, to this talk. The title of this talk is 3D printing for pharmaceutical and biomedical applications. And if I understood correctly, most of you are from either pharmacy or similar or allied fields. So what I'll do, I'll introduce this topic, that is 3D printing. Many of you probably heard this name 3D printing or maybe associated with this technology. Yeah. But I will start with very basic thing, that is what is 3D printing and then how we use this technology for either pharmaceutical or biomedical applications. Okay, so this is the outline of this presentation. So we'll start with 3D printing, general introduction. Then we'll see what are the scopes of 3D printing in pharmacy and also 3D printing for medical applications. So I'll discuss few of the examples and recent development in 3D bioprinting with this thing. So let's start with the very basic thing, that is what is 3D printing or additive manufacturing. Additive manufacturing is a scientific name of this technology, that is this 3D printing, and 3D printing is more general, like that's more general, and many people understand this technology by this name 3D printing. So basically, this is a technology by which you can generate a prototype from your idea. Suppose you thought of a particular design or a particular structure, a particular anything you can thought of, whatever, any physical form, anything that you can thought of, you can basically design that, model that, and print that or fabricate that into a structure, and then you can get this thing. So this technology allows you to get a physical prototype from a virtual prototype. Virtual means when you can design that in your computer using some CAD software. CAD is computer-aided design software, and then that design thing can be loaded to the 3D printer. 3D printer is nothing but a machine or an instrument that use this layer by layer manufacturing concept to print whatever your design is, that to print that thing into a physical form. So finally you will get a physical prototype out of it. So in 3D printing, if you can again divide this into 3 steps, like the first step is the modeling, where we generate the virtual blueprints of the structure, that is whatever you can design, you have the liberty to design any complex geometry using a CAD software, and then that design file can be loaded to the 3D printer, where the 3D printing is a layer by layer manufacturing process, where each layer is printed at a time, and then the next layer is again printed on top of that, and then simultaneously, or that thing keep on repeating every time to print the whole structure. So that is 3D printing. But after 3D printing, not all the printed products are market ready or user ready. So we need to go for finishing. So we're actually removing of support material or polishing, coloring, finishing, all these things, all these operations are done at the. So this is a 3-step process where we can get. Now we can print, nowadays we can print each and everything, whatever you can think. Suppose this is a skull model. So this is a plastic model, of course, there's no life to it, but this is a skull model, can be 3D printed and can be used. So similarly, we can print different structures. Suppose somebody's face, we can print a 3D printed face. so that we can understand, we can study very many things. So for medical, there are many applications such things, 3D printed of organs, 3D printed of tissues, all these are going on. And if you, the list is very long, so I have not shown here, but if you search, then you will get actually, with 3D printing, there is no limitation. Like we can print each and everything. Nowadays, we are printing tissues, organs, all these things, but still there is some challenges, but definitely we are printing few of the things, definitely these things are going on. And before even actually going to 3D printing, it is good to know 3D printing, is it a top-down approach or is a bottom-up approach? If you see, bottom-up means when there is nothing from the scratch, you keep adding the material and then you build something. So that's the bottom-up approach. And top-down means you have something already existing thing and you cut down into your required structure. That is top-down. So 3D printing is definitely a bottom-up approach, because additive manufacturing is a bottom-up approach, because there is nothing at the beginning, and then we keep on. It's an additive process. We keep on adding the materials, and then we are getting the whole structure. And 3D printing is not a single technology. There are many different technologies available within this domain, and each of these printing technologies have capabilities of printing one or different materials. So depending upon the materials, the processes are very different, and also depending on the processes, you have a whole bunch of 3D printing techniques. Like here, for when you work with thermoplastic polymers, then we can use this technology called fused deposition modeling, where this works on the principle of extrusion principle, and then we extrude the material and then we get the structure. Similarly, for different materials, like for granular material, we use this DLMS 3D printing technique, and also for metal powder and ceramic powder, we use this powder bed 3D printing techniques. So depending on the material, there are different techniques and there are recent developments. So I'm not going to details of all these things. This will be like too much details. So if you have any interest, maybe we can discuss later. But let me just go through why 3D printing is so important, and why 3D printing people have been adopting this technology for their applications. Like many of the pharmaceutical industry is little slow to adapt, but many of the research institutes and pharmacy colleges, actually, they are adopting this technology for developing different pharmaceutical dosage forms and also other in vitro tissue models. All these different researches are going on. I will give you glimpse of all these different technologies, that is all these different applications that can be possible with 3D printing. So the first thing is 3D printing. What is the application of 3D printing for pharmaceuticals, suppose? So you know very well what are drug delivery systems. So I need not to tell you about this, but just to tell you what are the advantage of current pharmaceutical drug delivery systems, available dosage forms, and what with 3D printing, what can be done, or where exactly 3D printing can be used. Suppose you know that 3D printing, drug delivery system is nothing but the method or process for administering pharmaceutical compound to a human body and maybe to animals to achieve a therapeutic effect, right? So suppose we are treating some disease or some disorder where we are administering some drug, so then we develop basically one particular type of drug delivery systems. Either it can be oral or it can be any type, parenterals or anything, like all these different types, and then we can administer that, right? But we know very well there is inter-individual variability. So all of us probably not need same doses of medicine all the time, right? That can be an inter-individual variability. But if you see, then most of the drugs come in a standard formulation, like suppose for paracetamol, it is standard 500 or 650 mg of paracetamol. It's a standard. So if whether you require 500 mg or not, but you need to, that is only available. There is no 450 gram or 550 grams of paracetamol tablets available. So that's so, but maybe that is required depending on, depending on the body weight, depending upon your body mass index. So there's the doses required may be varied. But with your current drug delivery systems, that cannot actually accommodate all this. The other thing is, if you want to come up with a new drug delivery system or novel dosage forms, then it is very hard for an industry to adopt that kind of thing, because they need to invest huge amount of they need to invest a lot of money to to come up with the supply chain, to come up with the production line, right? So because they invest huge on the production line, to if there is any change in the existing dosage form, right? So there is a huge cost associated with this thing. That's why adopting a very new technology is slow in industry. The other thing is developing new drug is always complex, expensive, and time consuming, right? Because that needs a lot of research. To develop a single novel drug, it takes years to come to market, and there's a lot of money involved, a lot of funding is required for that. So developing new drugs is very complex all the time. It's complex, expensive, time consuming. So that's why now what is going on is repurposing of some of the existing drug that is going on, right? But if what FDA actually encourages to develop more solid oral dosage form, okay, because that is very easy to administer. That patients also can use that very well. So that is one requirement where we need to work on solid oral dosage form. Right? The other thing is for industrial scale, a continuous process is very much important. So they work on bulk manufacture. They don't work on a personal or individual or personalized thing. That is it. But 3D printing is not meant for mass manufacturing. That's the very first precautionary statement. And the other thing is compared to conventional continuous process such as rotary press, that is very much used for this thing. The productivity of additive manufacturing is low, and the cost is also higher. And the production cost may not be paramount for personalized or patient-centric dose form. That's another thing. But one important thing to remember here is 3D print. When you are using 3D printing for pharmaceutical dosage forms, then the cost of that thing will be definitely a little higher than your standard bulk manufacturing process, like one of the rotary press and other thing. But if you are thinking about personalized dosage form, like suppose, as I said, to accommodate the inter-individual variability, then that cost is not very high, because 3D printing is capable of printing customized dosage form or customized form of any drug, right? So that is very much possible. So that's why 3D printing can be adapted where there is a specialized requirement, whenever there is a chance to improve any of the functions, any of the capability of a particular dosage form, then 3D printing can be very well used. So these are the advantage of 3D printing for pharmaceuticals. Suppose 3D printing can simplify the production process. That means with 3D printing, production of a novel drug dosage form is not very difficult. It's easy, and it's simplified. It's a one-step process basically. So that I will come to that. Why it is one-step process? What is the requirement of this? Another thing is with the requirement for active pharmaceutical ingredients also can be reduced, because when you actually make the dosage form with 3D printing, you need only that much amount of active ingredient that is required to fabricate the drug, not the whole thing. The other thing is also capability of producing printlets, like 3D printed tablets with a precise dose, tailored drug release. All these things are possible. So that's why 3D printing is can be advantageous for pharmaceuticals, where actually you can 3D print a tablet with precise dose, that accurately how much amount of is required, that much, and also tailored drug release. So 3D printing we can actually work on to come up with a delivery system where the release profile can be very much customized or tailored, right? So that kind of thing, that's kind of possibility, this thing. Other thing is combining multiple drugs into a polypill. So suppose one patient, he's taking 10 different medicines whole day. So it's a very complicated medication regimens, and it is very difficult to remember for patients. Like suppose this is, why can't we combine with all this into a single drug delivery systems? So that is like a polypill, and that can work on different, that can administer or that can release drugs at different time intervals, right? First one drug, suppose one drug will be released, the next drug will be released after a few hours. So that kind of things, why can't we do those things? So those are the advantages against for 3D printing for pharmaceuticals. And if you see the the growth of this market, like the growth of this technology in pharmacy, like 3D printed drugs market, it was like in 2018, it was here this much. But by 2025, it is expected that this will reach like almost $437 million market with a 7% growth. So this is a highly growing area, but 3D printing is more and more 3D printing is coming into pharmacy. And with all these are possible with 3D printing. Personalized medicine, I have given example of personalized medicine where one particular dosage form can be made for a particular individual, and then that can be given. So that can be prescribed, like the doctor can prescribe that this amount of certain drugs should be taken by this patient, and then somebody can actually 3D print that much amount of drug accurately, and then that can be administered, right? Similarly, personalized dosage form, like suppose for a particular patient, we need a particular type of dosage form. So that type of things can also be closed. Other thing is fast soluble dosage forms. I will show you one example. Fast soluble dosage form where actually one industry has introduced already, and that is FDA approved 3D printed drug for this fast soluble dosage regimen, where the drug release very fast, and for certain diseases, you know very well, for certain diseases that is very much required. Similarly, 3D printing can also be used to develop zero order drug release, where the drug release will be maintained. The concentration of the drug release from the medication that will be maintained over time. So there is no reduction in with the concentrated drug concentration in the dosage form, it is reduced. Then very often we see the drug release it is also reduced, but we can also develop a zero order drug release thing. The other thing is gastro-retentive protein tablets. They are another example where 3D printing can be very well used, because with 3D printing, one thing we can do, we can produce dosage form very lightweight dosage form. So those will not sink into the media or into the water. They will just float. Okay, so that kind of floating tablets also can be possible with 3D printing. Other than combined dosage form, as I already mentioned earlier, we can definitely combine different dosage form into one, and that can then. Other than that, in pediatric medicine, there is a huge scope of using this technology, because with pediatric medicine, we can work with form, color of this medicine, of the dosage form. So all these things are possible for with 3D printing for this thing. So these are the some examples where 3D printing can directly be used for developing dosage forms, but there are many other areas, and probably I have not listed those. And I encourage you to please go through the literature. We will come to know many of these things. Let me also point out here few of the industry, those are who are the early adopter of this technology, who has already worked on or already either introduced or in the line of introducing some 3D printed medicine to market for. Like the very first one is Apricia Pharmaceuticals. It's an US-based company, and they have come up with a 3D printed tablet called Spritum. Basically, the API is, you can read here, right? It's an anti-epileptic drug. So that this 3D printed tablet has some advantage. Later on, I have a video and I have some slides on this also. What is the advantage of this drug when you 3D print that drug into a dosage form with this company? So this Apricia is the very first adapter of this 3D printed, and also they have got US FDA approval for this thing. Now the GSK, they also partnered with Nottingham University in UK, and they have found that with inkjet printing and EV curing, they can produce 3D printed tablets, and that can be used for Parkinson's and for the drugs for Parkinson's disease. Like, so this is another example where 3D printing can be used for treating this kind of, like for developing drugs for Parkinson's disease. Merck is another leading science and technology company who has developed a partnership with additive manufacturing customized machines like AMCM to produce 3D printed medicines. Now, so that this is the trend now. Different pharma industry, they are either they are working with some industry, or they are another additive manufacturing industry, or also they are in partnership, they are working with some university to develop 3D printed medicines. So this is just 3 examples I have given, but the list is that's why I wrote many more. So there are many more such examples, and if you go through, you will find there are more and more pharma industry. They are working with some additive manufacturing industry or universities. So this is the most common 3D printing technology used for pharmacy. In pharmacy, because this technology can be directly used for pharmacy without any modification. That's why this is technology. Most of the pharmacists, they are working on this technology. Let me explain you this technology. This is a very simple technology where what happens. So suppose this is the print stage. So over this, all the printing operations will be done. So this is the stage build platform. We call it build platform. Over this, that 3D printing will be done. Now what we have a print head here. This is the print head. Right? This print head is attached to the XY movement controller. So that print head can move in XY anywhere. I can actually take this print head to with the help of guiding rods. Right? So this can move in XY direction. And what happens when it can actually basically it can produce droplets, right? I'll come to that, how it does it produce droplets, but it can produce droplets. Okay. Now, what we can do is, suppose we mix our APIs, active pharmaceutical ingredients, with excipients and all these things, and we make granules, right? So this granules, suppose we put into this on this build platform, right? So this platform, when you put this thing on this platform, and then now suppose with this inkjet head, as I said, this has capability of printing or generating droplets. If I load binder solution here, right, what will happen? Then I can spray binders, right? I can spray binder particles or binder spray. I can do that binder droplets I can always spray. Now I can put this binder droplets to a certain location. I have, suppose with this 3D printing, we have a control of that. With this, what I can do, we can control the positioning of these droplets at certain locations. So whenever we need the binder to go and bind these particles, we can basically drop this liquid drops. We can put this on that particular area. Now, suppose whenever we're putting these droplets, that time they are only the particles will get bind onto each other, or other place it will be not done. Now this, when the first layer is done, then build platform will lower be one layer lower. Then again this roller ball, it will level another level up, another layer of powder it will just put on top of this. Then the same process again, the binder will come, the printed will go and deliver these droplets to that particular location, and by this thing this process will be done. We have a video of this thing, same thing. Let us play the video of this thing. This video, actually there are many videos available on the YouTube, and then basically you can work on the same thing. We can see that. Let us see this video. Here if you see, these are the machine here. This is the CAD software, as I was telling you, where they are making different, like suppose you want to make this kind of structure. Now this is actually printing. This is the powder bed. Powder bed where actually you can put your granules here. Suppose you can also mix active pharmaceutical, some excipients, and this is the powder, and then you can just put this powder. So one layer of powder, suppose they are layering just one layer of powder on the stage. So this is how suppose the granules of powder particles that is getting layered. Right? One layer of powder particle is getting layered here. Also the compaction operation is done. Now the next thing is, this is the print head, and the print head is spraying binders. I said the binder solution, they can be loaded to that, and this is the droplets. Those droplets are coming down from the thing. So at a time, hundreds of droplets can be generated, and those can be delivered over the powder bed at certain location, not everywhere. The printhead can go and move print at that certain location. So this binder will bind the particles, right? And whenever there is no, suppose this green zone only where the binders is sprayed, the other area white zone the binders are not sprayed. So there the particles will not stick to each other. Only the green zones where the particles will stick to each other. Then after that, you just remove the powders from the. Again, this layer by layer they are doing it. So the level is lowered one layer. Again, the powder is loaded, and then again this process is done. This printing thing is done. So this is how the whole process is done. Right. So after this, if you just remove the material, then wherever the binder has actually bound the particles, those things will be together. Like this is the thing. This is because here the binder is together. Now after this, you can dry it, you can process it, you can curing, to do curing, different other things you can do, post-processing operations you can do, and then you can generate the structure, right? So this is how the binder jetting operations is done. This is very common now. Just remember this. Just replace this powder with that granules. You generally make granules for tablet manufacturing. So that granules, you can just put it over this and then do the same process again. So this will, you can get it directly, you can get it the dosage form, and you can produce any size, any particular size and geometry, you can do that. This is one example where, as I was saying, telling that today, yeah, this Apricia Pharmaceuticals, US-based pharma industry, they have developed this 3D printed spritum. It is an epileptic drug. So that's what. So let's see the video of their video. So where they have clearly shown, and this is, remember, this is a fast soluble drug. Fast soluble drug means where the drug has to be released very fast, right? So this will be solubilized very fast. It will be released very fast. Right. Sorry, just a minute. Yes. So this video, yeah, this is the video where you can clearly see that how fast that drug, how fast, you see, at 50% speed, whenever it is that touches water, it dissolves or dissolves immediately. It disintegrates into the solution and dissolves in the solution. This is the normal speed. So within a fraction of a second, it can dissolve. And this is the OTC fast melt drug that is available in the market, and this is the 3D printed drug. You can clearly see the difference here. In this case, the dispersion completes within 1 or 2 seconds, but here till now, you see, till 9 seconds, 10 seconds. So slowly the dispersion started, and then it will be there. So this 3D printed drug is outrightly outperform the OTC fast melt drug. So that's the advantage of 3D printing, where this can be, to develop a very high fast drug, that can be there. Now, why this 3D printed drug actually dissolving very fast? There is a reason for this, because if you see in our tablet manufacturing, it's a compaction process, so where we don't generate porous tablet, right? But with 3D printing, actually, we can create porous tablets. So that's so because of porosity, it can absorb water very fast and it can dissolve very fast, disintegrate with just 1 sip of liquid. So now think of this medicine. If whenever you put it on your mouth, just over the tongue, with the saliva, immediately to disintegrate and the absorption start immediately. So the drug will act very fast if you can use this 3D print technology. So this is the technology. So with this technology, they can better manipulate the drug composition and also how the drug will be delivered or administered. That can be very well studied, very well controlled with this technology. So that's the advantage of using 3D printing. So this is one advantage, where one example where 3D printing can be very advantageous for certain applications. But but please remember this: 3D printing is not for manufacturing all different types of drugs or tablets, or suppose tablets, capsules, everything. It is just whenever there is a very specialized requirement, that time only you can do it. We should do it. It is not for mass manufacturing. But there are some core challenges of adopting this technology for manufacturing of drugs, like developing a unique and innovative additive manufacturing system, especially for the pharmaceutical industry. Like most of the 3D printing or 3D printers, they are capable of printing different types of materials. They are not suitable for printing, like suppose very sensitive drugs or very sensitive main ingredients, right? So we need to come up with a technology where this technology can be used for manufacturing of, or we can actually customize a 3D printer for pharmaceutical applications. The other thing is regulatory requirements, because suppose this drug is a tightly controlled area, right? The regulatory requirement is huge. So that's why another challenge is how we can actually satisfy the regulatory body so that we can get the regulatory clearance. So that's why nowadays till now only probably 1 or 2 3D printed drug has been approved by the regulatory bodies, not more than that, right? Because one thing is, of course, the process should be safe. Like if the drug, wherever they produce the drug, that should be safe and effective. So those are the things one should remember. Now I will show you some of the applications how I 3D printing for for medical applications, where 3D printing is used for medical applications. One thing is, medical school people generally, they use some medical models like skeletal. Even pharmacy in pharmaceutical college also nowadays, many of these anatomical models like the skeletons and other things are there, right? The source of those things are mostly dead bodies, right? From there. But why can't we 3D print those skeletons? Suppose the 3D printed skeleton can be 3D printed, right? Is nowadays even various or tissue or organs can be 3D printed. So this 3D printed tissue or organs can be used as an anatomical model for practice, as a practice dummy, or for educational aid, also for as a preoperative planning, right? So suppose if I can print, let me switch to the, I can print even a, suppose I can print a hand. This is a hand structure, right? I can print a hand structure with all the bones, with the bones by separate color of material, ligament with separate color material, blood vessels with separate color things, even tendon and other things with separate color things. Then it is very easy for a student to understand how the hand, what are the structure present within the hand, and how does it function. So all these things very much can be very well done with 3D printing. So this kind of models can be very well thing. Other than this, suppose this is a 3D printed human brain, when all these lines, if you see these all lines are neurons that is connecting from one part of the brain to the another part. So if you can 3D print, like suppose this is a 3D printed one, we can clearly see when how this one part of the brain is connected to the other part by these neurons, right? So this kind of things can be very well done with 3D printing. The other thing is, this is another example where this is the Neurological Society, probably in Netherlands. What they have done, they have for practicing surgery, like this you know, the surgeons, they undergo practice, right? They undergo demonstration practice to improve the skill of surgery. So what they do, they don't do this surgery generally over dead bodies. They do over this 3D printers. So suppose this is a 3D printed brain, and most of the features of the brains are recapitulated when 3D printing of this thing, right? But these are non-viable, only the material, only made up of materials. There is no cells in there. So that kind of, now the surgeons can perform the surgery on this, and they can hone their skills. Right? So this is another example where 3D printed can be used for surgical simulation. So this simulation nowadays for pre-operative planning also, doctors they use 3D printing, because they suppose there is a very complicated cancer located at a certain region, right? Now doctors, before actually going for surgery, what they do, they actually ask to print this model of that thing, that's the particular thing, and then they do the surgery on top of this. How I should approach they have to remove the cancer, how I should approach it, what angle, what distance, all these things. So they do all these things before actually going for 3D print, before actually going for surgery. So these are also called pre-surgical models. Similarly, another area of 3D printing is the prosthesis and orthosis. Prosthesis, probably you know that prosthesis is when a device is used to replace or augment a missing body parts, that is prosthesis. And suppose if there is no missing body part, just to repair or augment a part, then we use some device, that is orthosis. This kind of, you must have well aware about the Jaipur foot, right? Jaipur foot is a prosthesis where somebody doesn't have a foot, then they can use this Jaipur foot, and then they can walk, they can all do all the other activities. But this Jaipur foot is mostly, it's an same for all, right? There is no customization. But nowadays Jaipur foot is also, they are also going for customization. So what happens, suppose somebody has one leg, the other leg, they can actually make a replica of the one leg. So basically it's a mirror image of the other leg they can make. With 3D printing, they can easily do that. And then that can help this kind of person to do their daily work. Like they can walk, they can do all other activities, right? Similarly, foot orthosis, like here, suppose somebody has a sprain or other thing, then one can actually 3D print this kind of sprain, and that can be very much customized. The most important thing is, suppose the shape and size of this human bodies, suppose the hand, leg, all these different organs, different limbs or different organs, they have a particular geometry, and everybody, there is inter-individual variability. So we can scan a particular, suppose we can scan a particular patient's leg, and then we can print a particular structure that can very well fit to that person's body, so that then they can actually basically use this for their daily life, and that can also help to heal the wound, that injury or that damage, right? So this kind of things are going on here. If you see conventionally, this is done just by taking impression. Like suppose there is a sand. Like now, suppose if you in the conventionally, people were being asked to put their, suppose I want to make something for the foot, so a foot orthosis, like, so they will be asked to put the foot on the powder, this bed where there is a, suppose sand is there, and then they will be and there will be impression will be taken. Now this impression, then there will be another, they will put some material to take the impression of this thing. So that is the negative replica. Now using this thing, then they will use it for making orthosis, putting materials on top of this, cutting, polishing, all these things. So this is a labor intensive process, and it takes time to do that. But nowadays, what is being done? Generally, suppose they can put feet on this thing, and that impression can be scanned. Similarly, person's feet or person's hands, that can also be scanned. Then when you scan that, then you can create the geometry. So you can create a geometry on using some computer program, and then that can be used to develop the virtual prototype, as I earlier mentioned, virtual prototype, and that virtual prototype can be used for 3D printing of the orthosis or prosthesis. Okay, so this process is very simple. One thing is very simple. This is a fast process. This takes less time. You know that the conventionally to manufacture a foot orthosis, it takes almost 2 to 3 weeks time. But with this kind of CAD, with scanning and CAD modeling and 3D printing, it can be developed within a week, right? Even within few days, it can be developed. So that's the capability of this 3D printing technology, where this can actually develop definitely. And the other thing is fit. It will perfectly fit to the person. Generally, conventionally, whatever they manufacture, those are not perfect fit. But with 3D printing, actually, the product that is developed, it is a perfect fit for that particular patient. And nowadays, 3D printing is used for all different applications. Here I have shown some like 3D printed braces, 3D printed cast. Like earlier, somebody break their bone, then what they do? They go to doctor just put a plaster of Paris cast. But nowadays, with 3D printing, what we do? We can actually scan patient's head, patient's hand, and then we can design a model that will perfectly fit to his hand, and then we can 3D print the structure. So these 3D printed casts are becoming popular, and probably in future we'll be using more and more this kind of 3D printed cast. 3D printed insole is another thing. You must have seen this in this Oscar Pistorius in the Blade Runner. He's also famous by the name of Blade Runner, because he doesn't have these legs, but he used these blades for thing. So in future also this kind of things. So a 3D printed, suppose 3D printed legs can be produced, and that can be used by the people, this thing. Okay, so all these different examples as I have shown here, they're just some glimpse of just few examples, but there are many more where 3D printing can be used to develop this 3D printing orthosis and prosthesis. So other area that I would like to show you is the 3D printing for reconstructive surgeries or for cosmetic surgery. Suppose somebody has a defect in their face anywhere, right? So with 3D printing, actually, we can actually repair, or we can rectify that defect, or we can basically so that people will now no longer feel isolated, or the social stigma can be gone. So that kind of thing. Let's listen to this thing, see this thing. Now, suppose somebody has a fractured a particular bone here. Suppose this jawbone is fractured, right? Suppose what you can do, we can actually scan, and then we can print. Like suppose we CT scan, we can get the whole geometry of patient's geometry. So then I understand this is the part that is missing, right? We can create this geometry just because there is a symmetry between left side and right side. So we can create, suppose this is the part that is missing, right? Basically, we can 3D print this part. Now, this will be a customized product for that patient. Also, we can include cells, we can include some certain factors that can help better integration of this structure with the patient defect geometry, and then it can be put back to that missing bone area. So this kind of thing is also possible with 3D printing, and this is This is just an example. There are many more this kind of things is possible with this 3D printing. This is for reconstructive surgery whenever there is a defect, and that defect can be because of various other. Suppose somebody made an injury accident, so that time maybe this bone is got fractured, and so suppose this is, and it cannot be, this bone is fractured into pieces. So then we have to, that area will be vacant. So we have to provide certain structure to there so that it can fill that gap. Similarly, so this is work from Prosthetic, South Korea, where this boy had cancer in his orbital cavity, below orbital cavity. Now this is the thing. Now because of this, his two eyes are not aligned. You see, one eye fall off, right? So this is, he has, because of this, I suppose whenever there is a defect or this thing, on our face, then because of face, then it is a problem, social problem, like problem with the person, because he will not be interacting with other people. He has a social stigma. He will not go out. Slowly his self-confidence will go down, right? But with 3D printing, actually we can rectify this problem. But if you get the CT data, this is a CT image, CT data, then we clearly understand what is the bony part that is missing, right? So this is the part that is missing, and we can actually basically understand that this is the part. We can actually extract that. This is the part that is missing, and just now we need to print this part only, right? So with 3D printing, actually we can print this part. This is the part that is the exact thing of that is the what is missing, and that then when you give it to the plastic surgeon, cosmetic surgeon, then they can put it back on the patient face. So suppose here, this is after 4 days, and this is probably after 2 weeks. So this boy is, this is probably after 4 weeks, and this is after 2 months, and this boy is doing fine now. So his eyes are again aligned on the same plane, and he is much more confident, right? Because he doesn't have any deformity in his face. So that kind of thing, this kind of thing can be done with 3D printing, where we can actually basically we can rectify any kind of damage on a particular tissue. The next thing is I would like to discuss, or I would like to introduce, that is the 3D printing for in vitro tissue organ model. And you know very well, the drug discovery process is a very long process. Huge cost is involved, and there are very less drugs that actually reaches market after all this thing. So what is the first thing? First thing, identifying some lead molecules, right? So when some lead molecules are identified, and then that goes through the 2D cell culture screening. When the cells are cultured and these drugs or these invented molecules, lead molecules, those are being tested here, and then some of them will pass. Those do not have any toxic effect. Others, they will fail because they may have toxic effect, right? Only these pass through the test. Then it will be tested on small animal model, like rat, mice, all these things. So here also, if the drug is killing the animal, then it is toxic. And if the drug is not then killing, then it is safe and for use, so that it will pass again. Some of this will pass, not all. Then it will be tested on a high animal model like monkey. And again, if it passes, then only very few passes this thing. And then it will be used on volunteers. And the volunteers, if the drug is toxic or not, that will be found in the volunteers. And then it will be used for common people to treat certain disease or disorder. But now this whole process, you see, the whole process is very long, and there are many steps involved. Many steps involved. Huge cost funding is required. Other than this, there is a ethical concern, because we are using animals, right? We are killing animals just for this. The other thing is, not all the time we get confirmed results. Many of the drugs, suppose they pass through this, all the process, but later on, when it is introduced to market, later it found that drug may have some toxicity. Then it is a big loss for that pharma industry, because they have to take that medicine again from market. They have to take it back. So that's why nowadays most of the regulatory body, they suggest to go for don't go for animal testing. Go for to use in vitro models, tissue models. Suppose if we can develop or design on the 3D printed structure, like now this thing, suppose this tissue like liver, kidney, or other any other organ, then definitely we can use them for testing drugs. So this is one area where huge amount of research is going on, and more and more industry, they are actually collaborating with for 3D printing industry for developing tissue organ models so that they can test their drugs. And please remember, please note that now European Union, they ban all cosmetics that is tested on animals. So you cannot use any cosmetic product or drug to test on that can be tested on animals and that can be marketed. It has to be tested on human models, right? So this is a one area like this thing. The advantage of tissue models are like you can do systematic study. Well, suppose you produce different types of tissues, right? So if you produce different types of tissues, basically they connect them together, and then you can go and do this different studies, like repetitive drug study can be done, and high throughput studies can also be done, like where the drug can be tested in a high throughput manner. So because you need not to kill animals here, you need to just produce this 3D printed tissues or organs in your lab, right? And then that can be tested. So you can produce hundreds of these tissue models, and you can test your drugs. All these different drugs can be tested. Even low dose of drugs can be tested. Combination of drugs can also be tested. You can do these things for understand the mechanism of action, to understand the drug response relationship. So all these things can be studied on tissue models. One such example is liver model, because liver is a very important organ, and you know very well wherever this drugs taken by oral route, that goes through the liver, right? So liver actually metabolize these drugs, and if the drugs have toxic effect, then definitely they will exert this on the liver. So liver toxicity of a drug is the primary concern for all oral doses, even for some other doses form also. So that's why the liver model is the very first one developed by industry for drug testing. So this is an example of how this liver model can be used. Now, suppose this is a 3D printing process where the cells are encapsulated within the hydrogel, and then these cells are printed into a particular structure, and then it is allowed, we allow that to generate the structure. Let's suppose the liver cells are printed here. This is the liver cells, printed and then cultured for some time. What will happen? The liver cell will they will proliferate, they will produce extracellular matrix, and then you can get a liver-like structures. Now, if you add some drug, can be tested on this, whether the drug has toxic, so the drug has toxic effect, then it will create toxins to the drug and the cells will die. Now, suppose some drug has hepatoprotective effect, so that can be also studied, right? Whether the drug has hepatoprotective effect or it is toxic to the liver, so those things can also be studied. Other thing is you are testing the drugs on 3D. In 3D, actually, the drug response is much more very close to the human body, because the human body is like the tissue is in 3 dimensional. The 3 dimensional tissue the response is very, very different from the cells are on 2 dimensional. So this kind of in vitro liver models are going. So this organoid industry, US-based industry, they have come up with, in 2015, they have come up with a in vitro liver model platform. Now they are giving this platform to various pharma industry for testing their drugs. Right now, so the application of this in vitro liver model is like all this, like to understand the physiology, pathophysiology, to understand the morphogenesis of the structure. Also, this can also be used for drug screening, like the different drugs can be, actually this model can be prepared and the different drugs can be tested on this thing, whether the drug has toxic effect or not, okay? And the developing infection model. In COVID time, this is very important. Suppose if you can generate lung model with 3D printing, then what will happen? Then actually we can inoculate this virus on the lung model, and then you can study how the virus actually is spreading on the tissue, how it actually infecting the cells, how it actually got into the cells. All these things, various things can be studied. Also, then when you can develop a model, this COVID infection model, then we can now test drugs, which drug actually acting on the model. So that way we can actually find out what are the drugs can have effect on for this COVID thing, right? So that kind of infection model is very important, and now researchers, they are trying more and more. Researchers are trying to develop this kind of infection model. The other thing is the in vitro cancer model. Suppose when if there is a cancer growing in the body, then mostly the treatment of cancer is like a trial and error. Like most of the time, because of the genetic variation, inter-individual variation, most of the patients, they don't respond in the same way to different drugs. So that's why we need to develop cancer models where we can work on collecting patient sample, isolating cells, use those cells to develop a model. So that model will be very much personalized for that patient. And then actually we can test what drugs can actually work on these models, so that we can understand this, so that then we know that, so these are the drugs that will definitely work on that patient. Not all the drugs will work. So that's this kind of models. So this kind of models are important. Right? Now the next set of slides are on 3D bioprinting, where 3D bioprinting is a relatively new concept. It started like in probably it is around 2010 or so. It started where the 3D printing is actually used for developing artificial tissues and organs. Then we can call them 3D bioprinting, where we use cells as a building material for 3D printing. Basically, where we use cells for printing tissues, that is 3D bioprinting. Like this is also layer by layer manufacturing process, similar to additive manufacturing or 3D printing that we have discussed. The difference here is we don't use any material that can damage the cells, that can kill the cells. So we use a matrix material that is very much biocompatible. Also, we use cells because cells are the building block of all tissues, right? So suppose I'm interested to develop a or to 3D printed liver, then I will be using liver cells like hepatocytes, cholangiocytes, endothelial cells, all the things I will be using, and then I will be printing the liver. Similarly, if I'm interested to print skin, then I will be using skin cells like fibroblasts, keratinocytes, all these things I will be using. And then I will also do one thing. I can basically print them at particular location, because tissue cells are located or positioned different at different locations, and there's a hierarchy, there is a particular way of arrangement. So all these things actually basically we can reproduce while doing 3D printing, and then we can come up with a functional tissue or organ. In this process, so there are 3 steps like pre-processing, processing, and post-processing. In pre-processing, what we do, we develop the 3D structure like, sorry? Okay, so with 3D printing, what we can do, when our interest is to print a particular tissue structure, and you know that most of all our tissues are very different from each other. And suppose the liver, in my body, the liver, and suppose somebody else, the liver will be very different. So we cannot print, actually, we cannot design the particular liver. This will be the liver for a particular patient. We have to take scans of that patient, and we do that with different imaging modalities. Suppose we can do 3D scan, like CT scan or MRI we can do, and then those scans can be used to develop the model, right? And that model can be used for 3D bioprinting. So that in the processing steps where actually we generate this bioinks. Bioink is nothing but 3D printable ink, right? Printable, where that consists of cells, matrix materials, biomolecules, all these different things are present within this thing, and then it can be used for 3D printing of tissue structure. So where cells are there, all these things are there, and then we just load this bioink into the printhead, and also we can load this model into the printhead, and then we print the structure. So when we just print the structure, we don't call them tissue; they are rather, we call them pre-tissue, because here the function is not yet fully established. These are only cluster of cells and materials, right? So then we allow them to mature, so that the whole tissue the tissue is matured further, the cell and the tissue regained its function, or the function is re-established, and then it can be used for implantation. Right now, again, 3D printing is also not a 3D bioprinting is not only also a single technology, depending upon the application, or depending upon the depending upon the operations or material being used, they are very different. We may have inkjet bioprinter, where it can generate droplets, and then the droplets generate droplets, and then it can be plotted on a surface or printed on a surface. Then it can be doing that. But here the Droplets can be generated either by heat thermal or by piezoelectric actuator, and then it can be, we can do this, we can actually then generate these droplets, and these droplets can be, we can get these droplets, and then these droplets can be used for generating any particular 3D structure. Right? So this is one such technology, the inkjet-based bioprinting, and this is very and this is one such technology where it is very similar to your office printer. Like where we in our offices also we have inkjet printers, where when you take print out, what do you see? We see it's the pattern of the black ink or coloring on the piece of paper. Either it can be image or text. Similar way, this also works in a similar way, but here the cells are loaded within the material, like suppose this buffer solution, and then the droplets are generated that can be plotted. In case of micro extrusion bioprinter, the cells are loaded in case of in the hydrogel, right? And then that is being pushed away through the nozzle, and then it can be plotted as filament can be come out, because here the viscosity is more, so there no droplets are generated, but here the lines are, this kind of filaments are generated, and then those filaments can be put into a print stage, and then you can generate droplet. In case of laser bioprinter, we use the help of laser, we generate droplets again, but from the bioink we generate droplets, and those droplets can be again plotted on the print stage, and this then we can get the whole structure. So this kind of these three types of bioprinting technologies are available. There are few more also, but these three are the most common one, right? Like we work on extrusion-based bioprinting, where I suppose in extrusion bioprinting, as I said, that we can either use some, means the typically a syringe is there. The syringe will be fastened to the printhead. So you can put any materials inside either cell-laden hydrogel or polymer solution, ceramic soil, everything can be put into together, and then we can push them either by pneumatic or piston driven pressure, and then we can generate the filament can be extruded, that filament can be put on the stage, right? So this kind of structure is possible with this kind of printing technology is possible. But when we use extrusion-based bioprinting, there is other challenges, because when you are using this solid material inside the syringe, that time it should be able to flow. But when it comes out, that time the material should arrest it, there should not be any shape distortion, or shape retention should be there, so that where solidification of the material or gelation of the material is a very good way to do that, right? Suppose when it comes out, if it solidifies, then you can get a very good structure. Otherwise, we can do various types of gelation mechanism we can use, like either you can use a cross-linking, photo cross-linking, chemical cross-linking, ionic interaction, all these different things are possible with this thing. Here, this is a video of extrusion-based bioprinting, where suppose through the nozzle how the material comes out, and then we can plot them into a structure. Sorry. Here, if you see here, the material is coming out through the nozzle, and that is being plotted on the print stage, right? And then again, in the next layer, another layer, suppose another material is, we can print on the next layer. Then the third layer, again, another material can be printed. So we can actually change the material, or we can also, even with the same material, we can print the whole structure, right? This is how the printing operation is done. Also now, suppose within this line, there can be cells, there can be biomolecules, all these things are also, so there can be drugs. Now suppose we can also encapsulate drugs within this line, and this can be used as a drug delivery platform, or also this can be used for, as a cells can be embedded within the structure, and this can be also used as a supporting structure. In this video, they have clearly shown how actually with 3D printing, how this organ bioprinting can be done. Like suppose, that for organ bioprinting, because all our tissues and organs, they are composed of different types of cells, right? So suppose we need 4 different types of cells to print a particular organ. So how we can do that? Suppose in your syringe, there are multiple syringes are there, and we can load multiple materials plus cells within this thing. And suppose the first syringe, suppose there are endothelial cells, like the vascular cells, and then there are muscle cells. Then there are suppose stem cells, 2 different types of stem cells are used. So suppose in this case, they are going to use 4 different types of cells, right? And also some material. Then one by one at a time, one by one, they will deposit one particular cells that is embedded in the hydrogel. Suppose that will be plotted or that will be printed in a particular pattern. So in this case, suppose a spherical pattern, the cells are being put together, put into on the plate in a spherical pattern. So one type of cells are, suppose they printed one particular type of cells, and then another type of cells are also printed simultaneously. So one by one at a time, we can print different cells at different locations, and also we can now, suppose our interest is to fill this venous structure with some material, so that can also be done. So this is one example, and then the next layer again. The next layer is also, or the same process is repeated again the next layer. So this layer by layer, we can build up the whole 3D geometry. Right? So this is how the bioprinting actually works to develop the artificial tissues and organs. So one nozzle will be working at a time, and then we can change the nozzle. Other nozzle can also come and work. So like this, we can actually work with the whole system. Another thing I would like to mention here is the bioink. Bioink is nothing but the bioprintable formulation where the cells, matrix materials, bioink, biomolecules, all these things are together. So bioink is nothing but a cell-laden hydrogel. It provides the function of this thing. It provides a necessary ambience to the encapsulated cells, because the cells, when you put them into the media, they into the hydrogel, they need oxygen, they need nutrition, all these things. So actually, when you are encapsulating this hydrogel actually providing them the nutrition, oxygen, all these things. The other thing is also this the hydrogel that also induce cells to mature and function, because maturation of the cell is very, very important. So this material or this hydrogel provides this necessary ambience. By this way, they also provide the suitable conditions so that the cells can function and mature. Right. The other thing is these are some key hydrogel properties that you need to remember. You need to work out when they go for bioink development. Like the very first thing is the biological part. The material or the bioink should be biocompatible. Material should be biocompatible and biofunctional. So it should not kill cells and should allow the cells to function. Now, the rheological point of view, it should have a sufficient viscosity, and also it should have a shear thinning property. It should have proper yield stress. And gelation mechanism, whatever gelation mechanism you have to use, either it can be, as I mentioned, it can be photogelation, or it can be chemical gelation, or it can be ionic interaction. All these things should be cytocompatible. So actually in our lab, we have developed a process where we work on, actually we have developed bioink directly from the tissue, where we collected the tissue, discarded human and animal tissues, and then we have developed a method by which we can convert them into a gel structure, because what we do, we throw out all the cells. So when we throw out all the cells, because the cells are mostly immunogenic nature in the tissue, and the materials are mostly they are not immunogenic. But so when we use this tissue, or when we use these tissues for developing this hydrogel throughout the cells, we use only the matrix material, we convert them into a gel, and then also encapsulate cells within this. So load the cells within this, mix the cells within the hydrogel, and then we print the whole structure. So in this way, this is a cellular construct. Then what will happen? The cells will grow, they will proliferate, they will migrate within the structure, they will produce extracellular matrix. That means the tissue building process will start happening, and then we can use this thing for either for implantation or for different other applications. Now I have come to the end of this talk, where this is the question, probably many of you have these questions, that when we can get these 3D bioprinted tissues and organs, right? Because with bioprinting, there's a huge possibility to actually develop artificial tissues and organs. But if you see, there is a huge gap between the demand and supply of this thing. What is the replacement of tissues and organs available? Like suppose somebody's liver got failed, or kidney got failed, then he can only have a transplant. Suppose if somebody is donating a liver or thing, then only he can get that thing, and that can be transplant back to his body, and that he can survive. So basically here we are depending on organ donation. But why can't we actually 3D print a tissues or organs, and that can save many patients? Because if you remember, every 30 seconds there is a patient dying because he or she waiting for a transplant tissue and organs. So that kind of things we can actually basically we can minimize that, and we can actually supply this artificial tissue and organs to many patients, so that can by this we can save the patient's life. But see, like in two-dimensional, some two-dimensional hollow tubes can be developed very fast, but hollow organs it will take time, and solid organs definitely it will take a lot of time. So this is the realistic picture where 2 to 5 years we can get this tissues and organs. We can get another 10 years we will be able to get this kind of tissue structures, but probably another 2. But if you are thinking of a full organ like liver, whole organ like liver, kidney, and heart, all these things will take time. So maybe at least next 15 or 20 years, maybe we can be able to do that thing. So let me acknowledge many people here. I have been collaborating with many people for different works in this domain. We have been working on in our lab, mostly I work on 3D printing for biomedical application. I don't do any work on pharmaceuticals. Currently, I'm not doing anything. But if any of you are interested, definitely we can start collaborating, and we can do that also. And these are some of the students that they have been working in my lab for some time. And I'm also fortunate to get funding from different funding agencies like SARB, DST, DBT, MHRD, ICMR. Also, recently we have got funding from one private funding, that is CPVF, for our cornea hydrogel clinical translation. So with this kind of things, this thing. And also I have actively collaborating with many people in India, basically from University Institute, and also abroad, many places. So I thank you all for your listening, and also if we can discuss now, because we have some time, I think we can take a few questions. So I'll be happy to answer any questions if you have this time. Thank you. And also I would like to thank the organizer for providing this opportunity to me.
Okay, thank you, Dr. Falguni. And actually, so one participant has a question.
Yes, please.
Any companies are there for large scale production for 3D printing? So Mr. Ravi Kumar is asking,
yes,
any companies are there for large scale 3D printing?
Large scale 3D print for what applications, if you can clarify?
Maybe like for bone implants, something like that.
No, actually, as I said, this adaptation of this technology to biomedical industry is a little slow. So now there are a few startups who are working on but now big 3D printing industry, they are also coming into picture. They are also trying to build this kind of things.
Okay. So actually, I have
I'm sorry, but for bone, actually, there are like for bone there are this metallic implants, even ceramic implants. Those are some of these things are 3D printed, and those are being differently. That is some industry they are doing it.
Okay. So actually, I have a question, and before asking, you have cleared me.
Yeah.
So when the tissue and organs are available for transplants, actually, so liver and heart and kidneys and all. So here you gave that answer. So it may take another 10 to 15 years, something like that.
Yes.
So
for a fully functional organ, definitely it will take time.
Yeah. Okay. So okay, sir, you gave the wonderful elaborated presentation. So very nice. So I would like to give any questions. Anybody else? Any questions? Any participants? If you want to ask any questions, please ask.
Also, in the last slide, I have given my
Yes.
email ID.
Yes, sir.
So you can also, suppose if you are not able to ask any questions, so if you want to collaborate, if you want to work with me, or if you have any questions or comments, size and anything, you can write to me. My email ID is falguni@bme.ith.ac.in. You can search Falguni Pati, IIT Hyderabad, you will get my profile. You can write to me. You can drop anything.
Okay, okay, sir.
Yeah.
So with this, I'll give some concluding remarks.
Yes.
So once again, thank you very much, Dr. Falguni, for your wonderful presentation. And with this presentation, I got some idea about how actually 3D printing works, and also how the technology of 3D printing and what are the use of 3D printing. And Dr. Falguni in his presentation explained about the scope of 3D printing and additive manufacturing like modeling, printing, and finishing. So the 3D bone implant and ear cartridges. So how they are made and what are the in several applications. So evolution of 3D printing and 3D printing in pharmacy. So has explained about fast dissolving, how the fast dissolving is made by the company. So advantage of 3D printing in for pharmaceuticals, and has explained about the technology used in the 3D printing, the material and binder jetting technology, and also has shown a video about 3D printing technology, which is very useful for the viewers. So which is very easily understandable, and also has mentioned a company which manufactures the fast dissolving tablet, like maybe Applicia, and what are the core challenges in the 3D printing, and and also has focused on 3D printing for medical applications. So I think this is most useful in the education and all, as well as for example, he has given anatomical models and practicing surgeries. So for practicing surgeries, so this technology may help to improve the skills of the surgeons.
Yes.
And also, he has mentioned about the 3D printed prosthesis or orthosis, like foot prosthesis and foot orthosis. So based on the customized patient needs. So based on the customized patient needs, they can make the prosthesis or artificial limbs. And he has mentioned about the reconstruction of bones and facial, especially facial reconstruction. And he has explained about the in vitro tissues and organ models. And also, he has discussed on drawbacks of animal models in the drug testing. And also, he has mentioned about the company Orthonovo liver tissues, so who has developed the liver tissues in 2015, and 3D bioprinting, and also has mentioned about the bio inks. What are the bio inks and and also has mentioned what are the different technologies used for the printing process. So the inkjet printing, extrusion printing, and laser printing has mentioned those. So these are all very useful for the viewers, sir. So thank you very much. Thank you very much for your presentation.
Yeah, you're most welcome. Thank you.
Thank you so much, sir, for
your
excellent presentation.
Also, thank you all the participants for their attending. So I have already mentioned my email ID.
Okay,
It's a chat box. I dropped that my email ID. Anybody has any questions, comments, you can write to me.
Thank you, sir. Thank you.
Thank you very much. Then, yeah,
thank you.
I'll leave the call.
Okay. With this, the session is concluded.
Dear participants, tomorrow we have the session, 7th session, that is from Dr. R.K. Sareen, sir, Professor, National Forensic Science University from Goa. So the session will be starting at 10:30 a.m. as usual. As it is mentioned in the program schedule, there's no change. Tomorrow morning, 10:30 will be. Okay. Thank you. Thank you.
Okay, Dr. Rifan. Thank you.